Hey everybody, so welcome to another video for the iTrack DNA project. Living Lakes here on site with Jared Hobbs, and we're at a Lotic site to study
giant salamanders. So this site, what do you think, Reagan? I like this site. I have some reservations, but what I like about it is we've got some pocket pools, and giant sals really like those. They tend to hide along the banks in those overhanging areas along the bank edges, and they tend to use the pools. I think we should see eDNA concentrations in the pool and then flowing between in the thalweg, so that gives us nice transport. In terms of the timing, we're good pretty much any time of the year. This is a slow-growing amphibian. They take up to 4 to 6 years to grow in these streams. The eggs are laid in the stream, so there's a permanent source of output. I think this is actually a pretty good site, with the exception of my reservations about it being ephemeral, but there's only one way to find out, and that's eDNA. So what do you think of collecting there?
The thalweg looks great, and the site sounds perfect for giant salamander eDNA, so let's do it.
Let's do it. And you know what's neat about this? Is there's not often you get to say thalweg in a sentence. So let's do it. So what do you think of this thalweg? It looks pretty good?
This looks like a great spot.
All right. Do you mind just labeling my bubbles for me? Is that cool? So this is Enchanted Creek. Let's call it EC1. And that gives us a chance to collect upstream afterwards at EC2 kind of thing. So EC1A for field replicate A. The collection time we've got is 1830, so that's perfect. And the initial, you've got my initials down. That's wonderful. So that's perfect.
Already.
Wonderful. So I'm going to do my rinse. To do the rinse, we want to make sure that we're getting all of the residual bleach out, because last night when we bleached these and cleaned them, we might have left some bleach in, even though we rinsed them in the bathtub properly. And so I'm going to do a triple rinse on site, and I want to do my rinse downstream from where we're going to be collecting so it doesn't influence things. I'm also, when I do my rinse, I'm going to be chucking the water out and away.
So do you want the collection pole for this site?
Actually, because we're at a Lotic site, I'm going to use gloves. The collection pole is awesome for lentic sites because we can get some separation, but here we're in tight spaces, so I'm going to be having my hand right in. So I'm going to put on a glove, and we're good. I'm not going to glove up this hand if that's okay. These gloves are a little tight, but we're good. So here we go. So once you've got the glove on, you got to remember that this is, in theory, clean. So I'm not reaching into my pocket with it to get Sharpies. I'm not touching our packs, that kind of stuff. So here we go. All right, so this is just the rinse to get rid of the bleach. So about a third full, good vigorous shake. Chuck it on bank because I don't want any of that bleach water going back into the system. We just saw a Pacific chorus frog in here. We don't want to affect him at all. So there we go. So that's our triple rinse. And now we're just gonna fill with water from the thalweg. This is a great spot. That's super convenient. And we'll check it to the top down.
Wow, lots of tannins.
It's lots of tannins. Good observation. So this is going to be hard to filter. We're probably only going to get like 300 mil through, but we're going to do our best, and that's just a consequence of the environment. So there we go. So let's put this on the shade just there. We're sheltered enough, and we can do our next bottle. But before we do that, why don't we collect some data about the site? And I'll just think about the tannins and everything else. So what do you have for field form or for field data here?
So we're at Enchanted Forest 01.
Yep, sounds good.
We're at Sample A.
Yep.
We're collected by Jared Hobbs.
Okay, perfect.
Date, April 28th.
Yep.
And time is 1830.
Perfect.
UTM.
Yep, I'll grab my phone and do that. So I'm going to do that at the end, and I'll get you that. And what we'll do is we'll take a picture using an app called Theodolite, and it actually imprints the UTM right on the picture, and the gradient and everything else that we need. So
and
that's
elevation?
Good. Yep, it'll note the elevation as well.
Great. Okay, so how about some sampling conditions, like precipitation? None.
Yeah, we haven't had any rain here for about 4 days, so we're in good conditions.
Great. Cloud cover, 0.
0. It's been a nice, dry, warm day.
Air temp.
Air temp's about 18. I checked it at the truck before we came down.
And water temp.
The water, we've got a thermometer, but it's back at the truck. And so we'll go collect that because we're parked right beside the stream. We'll collect that before we go. So just remind me. Thanks.
Perfect. Habitat description we're entering into now. So stream order. We said S6.
Yeah, so this is really important because this will help us when we're trying to evaluate or interpret the data after, and we can consider the suitability. So with this one, it's less than a meter across. I think it's safely an S6. If you could make a note, we both suspect it might actually be ephemeral. And so let's just make note that it's an S6, possibly ephemeral.
Got it.
And if it is ephemeral, that's going to invalidate it for Pacific giants or for Pacific giant salamander. So we're good.
Okay, reach description. So pool, riffle, run, glide.
Yeah, so we're sampling in between two pools, two pocket pools specifically, and we just collected in a little bit of a run here.
Okay. And then the dominant substrate.
Dominant substrate on this is cobble, but there are some fines. So it's cobble and fines.
Okay.
Not much for interstitial
put, like,
spaces.
a percent are those roughly?
No, I think you could say, let's go with that. Let's go with 30% cobble and 70% fines.
Subdominant substrate.
Subdominant substrate would be muck.
Okay, and embeddedness.
Embeddedness is fairly high, so I'm not loving this. For tailed frog, it'd be terrible. For giant salamander, I could see them pulling it off, but they're really going to be relying on bank overhang. So yeah. Okay, what's up?
So high.
Yep.
Gradient.
Gradient is low. This is like a 10% at best.
Okay. Channel stability.
It's very high. So to assess channel stability, I basically am looking at the banks. And if there's a lot of extreme flooding event that blow the whole creek out, then you won't see vegetation down the banks. You won't see bits of coarse woody debris and fine woody debris. So this indicates to me that this doesn't receive big floods. So the channel bank stability is high.
Great. And do you know if there's any logging activity in this area?
There is just upstream from us, about 300 meters. There was a clear cut. So
How long ago?
it was about pole sapling, so probably 10 years old.
Okay. And riparian cover?
Riparian cover at our specific site is actually pretty good. We're looking at about a 50% crown closure or so. So
Okay.
it's looking pretty reasonable, 50 to 60.
And riparian width.
Riparian width on this is easily 50 meters on either side, which is infinity to the giant salamander, so that's good enough. So we got 50 meters of riparian.
And the field-based suitability rating.
I would give this a moderate. I don't love the substrate, and I don't like the muck component. And I'm concerned about the potential ephemeral nature of the stream. So I would give this a moderate. What do you think of that?
I think that's really good reasoning.
Okay, sounds good.
Will we come back later in the season to see if it's ephemeral?
Yeah, that'd be a good idea to come back and check it in August and see if it's dried out at the lowest time. We can also, depending on what the eDNA results are, we can use that to dictate if we want to come back or not. If we get something in it, we might want to check again, because if it's ephemeral, we shouldn't. So.
Great. So our photo ID will be on the Theolite app?
Yep, that's right.
And did we observe any fish?
I did not. We did see a Pacific chorus frog, so Pseudacris fragila, or PSRE is the species code. And we haven't seen any giant salamanders. We haven't really looked.
No tadpoles, no adults. Any comments?
No, I think it's good. I'm just concerned about the ephemeral nature of things. So, all right, so yeah, I just want to we collected our sample. I feel good about that. We got the data for them, so we just have to get the picture with theodolite. And I just want to show you on the GIS app we are in the right spot, so we're exactly where we need to be. And you can see that clear cut just upstream from here.
That's great.
Yeah, so I think our comments are solid. And now I'm just going to bring up the theodolite app and just get those pictures that we talked about.
Great.
So yeah. So this one's nice. You see how it notes on your picture when you take it. It's actually going to include the UTM, the elevation, the gradient, everything. And I can give this a name and just call it EC1A. And then that's my downstream shot. And then I like to get the habitat and the stream and the substrate upstream as well.
Great.
So I'll just go ahead and shoot that. And those are both going to be time and date stamped and have everything on it. So I think we're all good here and we're ready to go back to the field lab, have some dinner, and then get filtering tonight.
Let's go filter.
Awesome. Cool.